So good morning, everybody. My name is David Bonifacic, and I am the CEO and one of the founders of WB Engineering. I am proud to say that this will be our WB's 25th year in business. And WB started in the dot-com era designing data centers, but today we're here to talk about clean rooms. So let me just start a little bit with a little data about the clean room market. In 2022, the global clean room facility market was valued at about $60 billion. And it is expected to reach over $100 billion by the year 2028. This is definitely a market that is expected to grow about 10% a year. For many, the first thought we think of when we say clean rooms, we envision people wearing those white bunny suits, and it's a space where they make pharmaceuticals and computer chips. But as Ray and Drew will discuss, clean rooms are also used in more than 30 different industries, including aerospace, defense, and of course, semiconductor. And that's why we're here today, because this is a growing industry, and there's some important things to keep in mind about the design of these facilities. So I'd like to introduce the speakers and panelists. First, Ray Doyle. Ray is a WB's Managing Director of Science and Technology. Ray came to us about 15 years ago, and he said he wanted to work on science and technology projects. And in 15 years, Ray has built a very successful national practice for us. Ray first started out of our DC office, but now he's based out of Raleigh, North Carolina. And one of the things I admire most about Ray is that he strongly believes in mentoring and teaching internally to our staff. He takes this part of the job extremely seriously. And one of the WB team members that Ray has mentored is Drew Zahratka, one of our other panelists. And let me tell you a little bit about Drew. Drew is a senior project manager and senior associate. He started with us in our New York office. He started as an intern, actually, in our New York office, started his career in our DC office, and now is with Ray in our Raleigh office. So I personally would like to say he's one of our up-and-coming stars at the firm. His experience includes managing projects for clients, including AstraZeneca, Catalent, and Kite Pharma. So before I hand it off to our panelists, I'd just like to say we really enjoy having questions. Please put all your questions into the question box on the Zoom app. And at the end of 11:40 Eastern time, we're going to open it up to an open discussion, but we will try to answer the questions as they come. So with that, I'm going to hand it off to Ray and Drew.
Thank you, Dave, for that great introduction, and thank you everybody for joining. So we'll just start with the real quick. Why are we here? Why do you really need a clean room or a clean space? And as Dave mentioned, there's a lot of industries that use clean rooms and clean spaces, and it really comes down to the need to monitor and measure and control particles down to the size and scale of a micron. The real context there is that most people can't see a micron. So if we move forward to, yeah, there you go. Thank you, Drew. 1 micron is also equal to 1 micrometer. So in various industries, you'll hear those terms used interchangeably. So again, to give you some context to that, an edge of a piece of paper is 100 microns. The average human hair is between 70 and 80 microns. But then you start to get into what really is hard to see, right? So if you talk about 40 microns is fine dust on the floor, you start to then go from that point of reference on the left side to a point of reference of 3 microns. When you start to get the things you can't see, everybody's used to the images of COVID during the whole COVID period, which hopefully we're long out of. But the idea being is you can't see that, and it does carry and float in the air. Talcum powder, white blood cells, and red blood cells are all in that same category of things you can't see.
So what are clean rooms used for? Dave mentioned, there's a bunch of industries out there that use clean rooms. One of them that would be kind of touching in a little bit is aerospace. So in the aerospace and defense industry, you'll use clean rooms for spacecraft and satellite manufacturing, microchip and sensor development, and manufacturing of engine components. In the food science industry, clean rooms are used to develop and research new seeds and pesticides. as well as growing plants. In the medical industry, you'll have clean rooms. Your clean rooms are your operating rooms, the hospital pharmacies, and even patient rooms for immunocompromised patients. Medical electronics industry has clean rooms for med device production and packaging. In microelectronics, you have clean rooms for semiconductor production. You have electric static dissipation clean rooms, battery dry rooms as well. In pharma, you've got manufacturing plants for your clean rooms, and then as well as for cell and gene therapies. And then quantum computing. This is one of the up and coming industries that we're really seeing a lot of boom in, where you need clean rooms due to the sensitivity of the lasers and the science that's going on there. Dave mentioned the growth of clean rooms, so there's a big push to bring manufacturing back to America. The 2022 CHIPS Act, for instance, is one of those acts that is putting into money to bring in manufacturing. Back in 1990, the U.S. had 37% stake in all of the world's chips that were being produced, whereas today we only have 12% stake. So again, there's a big push for manufacturing back. It's going to require these clean rooms for these advanced tech industries. The first part of our presentation, we're going to go over designing clean rooms. So the most important part when designing a clean room is getting that meeting at the beginning of the project with the end user. It's imperative to get with the end user to understand what are their protocols that they're doing and what are the engineered solutions that they look for in order to maintain the clean room. We're going to touch later in this presentation on the protocols put into place to maintain and operate the clean rooms to keep them clean. But during this portion, we're going to go over various engineered solutions and design solutions to maintain a clean room. So first and foremost, a clean room is quantified by the amount of contaminants at certain particle sizes. So Ray mentioned down to the micron, and in order to filter out particles of that small of a size, we need to use HEPA filters. So HEPA filters have a 99.97% efficiency down to 0.3 microns. Because the HEPA filters can filter out such small particles, we need to take out the bigger particles ahead of that. So you'll use a pre-filter, you'll use an intermediate filter, which is like a MERV 13, MERV 14 filter, and then you've got your final filter, which will be your HEPA filter. The HEPA filter can be located either at the air handling unit, it can be located in the diffuser, or it can be located in the room via a fan-assisted HEPA filter or fan filter units. So HEPA filters also have a high pressure drop, so it's very important to understand the total pressure drop when designing your new air handling unit, understanding what's the external static and accounting for that when sizing your supply fan and potentially putting in a return fan if you have a recirculated room to assist with the incoming pressure of the air handling unit. When retrofitting air handling units to reuse in clean rooms, possibly due to lead times and needing to get into the space quicker, you might have to reuse an existing piece of infrastructure that's in the room. So it's important to try and understand what's the external static and capacities of that fan. And if it doesn't have the additional external static, you can utilize a fan filter unit on the supply side to still get that HEPA level filtration.
And Drew, could we have some questions here?
Yep.
So one of the questions, is there a difference in how different particles are treated and managed within the systems, such as viral biologics? So can you go back to that slide with the filters?
Yep.
So viral versus biologic versus dust and sediment. I know you kind of went through this quick, but in respect to with COVID, you heard a lot of these terminologies being used in respect. So I think the question of viral and biologic as it relates to here, can you maybe answer that?
Yep. So as in terms of viral and biologics, some of those spaces, depending on what the contaminant is, you would exhaust directly out of the room, so there's no recirc back to the space. So you can have, depending on what's the risk in the room, you may not have a recirculated air handling unit due to potential cross contamination. So you would have a once through space. But then in terms of sediment and dust, that's where you've got your pre-filter to pick up the bigger pieces, and then down to the HEPA filter to get the finer molecules and particles within the airstream.
So you mentioned something that was interesting. Recirc versus 100% outside air or outside air. How do you determine when you can just recirc and when you can take from the 100% outside air?
So that's going to be something that's you need to establish with the end user at the beginning of the project, understanding what the science within the space is. And again, if there's any risks with contaminants due to the science in the space, and the end users typically give that requirement on what they need because of the chemicals they may be using, or the exposure that's there. Whether it's allowed to be recirculated within itself, it can be recirculated and shared with adjacent spaces, or if it needs to be once through air where air comes in and then it's exhausted out of the building.
So when you say end user, it's actually the person who's going to be working at that lab.
Correct.
Okay, so everyone understands.
In a lot of cases, Dave, you would consult a toxicologist or an EH&S or the safety person. Sometimes the user themselves that's in the lab may not have the expertise to know when it should be one or the other, whether it's 100% outside air or recirculated. So there are certain cases where you work with toxic or hazardous chemicals where they may want to consult an additional specialist to make sure that they are applying the right strategy to the airflow, recirculated or 100% outside air.
Okay, thanks.
So,
all right. So kind of at a high level, we'll start with some basics just to talk about how you design a clean room. But the first thing, as you start into the design of a clean room, you need to establish some kind of general vocabulary, and that starts with airflow and how it moves through the space. So there's laminar airflow, which is on the left. Everything's moving in a direction. Everything's moving together. Temperature, humidities, and flows are all going in one direction. And then there's turbulent airflow, where the theory is that everything mixes, and you can make the assumption in a turbulent model that the temperature and particles can move anywhere in the space, which obviously is uncontrollable. If we jump to the next kind of mindset in clean rooms, you'll hear the term unidirectional airflow used all the time when you talk about clean rooms or clean spaces in all industries. And it's pretty easy to understand, but you really got to have a nice little diagram to depict how it works. So if you're moving from left to right, it's going really from one wall to the next. And if you're going from top to bottom, you're really going from the ceiling to the floor. And the reason that's important is that's a true unidirectional flow model. And if you jump to the next one for me there, Drew.
Yep, just real quick with the unidirectional from top to bottom, you'll usually have a raised access floor in this instance. So it's going through the floor, and then it'll pan out to the walls below the room volume.
And then the HVAC takes over and ducts it wherever it needs to go. And then in the case where it's probably more commonly seen as non-unidirectional flow, where you'd have ceiling HEPA filters and it would go out through a low wall return. A lot of times people misuse this term in the case of science because they like to call it a unidirectional flow, but it's not a true unidirectional flow. It's a unidirectional flow at the top of the room and maybe down to a certain profile in the room, but then it becomes turbulent at the lower levels before it draws out the low returns. So it's important when we talk about these things, we use the terms correctly. Same thing on the right, when you feed from the bottom and feed to the top, you can get a good flow from left to right, but there's a little more turbulence across the occupied space or the equipment within the space.
Ray, can you give us some examples of where you would apply, in your experience, where this has been applied?
No, that's good, Dave. So in microelectronics, clean room, chip manufacturing, subfab spaces, you'll have a true unidirectional flow where they have everything coming from the ceiling, and they'll have a perforated raised floor across the entire floor, and then all the air goes down through. And so you get this, it's unidirectional flow. It's only ideal in a steady state and I'll talk about that in a minute, but when the space isn't occupied, people aren't moving around. As soon as you have people moving around, there becomes a little bit of turbulence. But the intent is, at a steady state, that airflow is going to go straight down through the floor. We've done the same thing in some aerospace applications where you have a huge high bay space, and we'll talk about this again later, where we have a sidewall condition where it's left to right, where they'll have large satellites or equipment in these large spaces, and the air will flow from left to right and then go out the other side, similar to the previous slide. So, and then in most cases in the world of biotech, until you get up into the ranges, beyond ISO 6 and you get into, the EU of classes around into the B and C ranges where you have to have a true unidirectional flow and get better airflows and better control over the airflow. So depending on the industry, Dave, you get different applications, and that's really what it comes down to, is how specific does the airflow need to be at a specific point in the space, whether that's horizontally or vertically, to either protect the equipment or the science that's being performed.
So Ray, I just want to emphasize, someone also asked along the same lines, what are examples of use of unidirectional Model 1 left to right?
Yeah, so the left to right one is specifically the applications that I've done it in have been aerospace. There may be
Aerospace.
other food science applications, but I find that to be most applied in left to right unidirectional flow when you have aerospace applications that need really high levels of clean room class at the HEPA filter wall. And in those cases, we'll talk about one later because I'll give you another example in a few minutes where they actually staged it across the room.
Right. And along those lines, does clean rooms, another question, do clean rooms recirculate air for energy efficiency reasons, or is it always supplied by fresh air intake? I think you answered that before.
In most spaces, yeah, it's really a toxicology issue. In most clean room spaces, it's large recirculation. It's really in pharma or chemistry use or toxic use where you'd want to exhaust that out. And usually when you exhaust that out, you're going to exhaust it out to the atmosphere, out of the building through another containment filter system to protect the neighborhood when you exhaust it out if it's a highly toxic space. So I don't want people to think you're just dumping it out of the building.
And to touch on just the energy, there is more energy that goes into it when you use 100% outside air, because you need to condition the air from more extremes versus recirculating. You're already starting at a better, more desirable room condition.
Right. And along those lines, Ray, when you said the toxic, like those filters, I mean, there was a question here, how is the different particles, how they treated and managed within the systems? So all those filters that have to be changed that are, how do you, maybe you can answer it later, but how do you deal with all that?
Yeah, the maintenance and changing of the filters is a real important aspect when it comes to downtime of the equipment. And we can talk about a little bit more in the operation, but the main point there is understand how dirty your space is. There's clean rooms that are very clean based on what's done in the space, and then there's clean rooms that have dirty activities. I'll use like tablets or pellets or equipment. If you're making pharmaceutical tablets, it's a dusty process, and it still needs to be done in a clean room environment. So sometimes that exhaust can go back to the air handling units and have a lot of dust in it in the exhaust. And even when you recirculate it, you may need to have pre-filters on that exhaust to protect the ductwork. So there's a lot of different applications. To answer that question, we might want to get together with that person individually and make sure we understand the application. We could better answer that question.
Okay, thanks.
So then the next thing we talk about the design tools for, and basically when we're trying to design clean rooms, we use different tools. We talked about different ways to lay out airflows and different HEPA filters in different locations. But the next tool that sometimes is applied is CFD, which is computational fluid dynamics. And it's not applied everywhere and sometimes misapplied. But in this particular image, you can see how it's important to understand that the HEPA filtered air blows straight down over a table, and that creates the cleanest environment in the space. And if all you're worried about is that one zone within the room, it's a proof of concept. It verifies that based on where the returns are and where the supplies are, that air is not going to recirculate back on top of itself in the room. So CFD does a great job of proving that design concept. In some cases, it's less important, and it's more important to just make sure you use good standard practices and use particle counters to make sure you're doing things correctly.
So now that we talked about how to filter the air and kind of how air moves through the space, the next thing in design is controlling the pressure. So utilizing pressure differentials will help control contaminants flowing into your clean room. So you would do this by pressurizing one room via a volumetric offset to the adjacent rooms, and then maintaining a differential pressure across the door. And this is all designed in a closed door um, arrangement. Some clean rooms, which is what we've got up here, as you can see, has an anteroom, which is an airlock that is used as another layer or level of separation from your dirty corridor, or kind of where common foot traffic is. So in order to get into the cleanest space, you have to go through another level of another room or level of different pressurization. Um, your anterooms can be designed as sinks, bubbles, or they can have a standard pressure cascade. So a standard pressure cascade is when the air flows in through the room and then out through the other door. Your sink is when that room is completely exhausted and air will come from the clean room and from the adjacent dirty corridor into the anteroom and then get exhausted out. And then a bubble is when you pressurize your anteroom so that nothing can flow into the anteroom from the dirty corridor and nothing flows into the anteroom from your sink, from your clean room.
So one of the things that is often misunderstood in clean rooms and in pressurized spaces in general, but it does cause problems in clean rooms, is the fact that moisture does travel in the opposite direction from pressure. So if you get a moisture, we call it moisture migration, but it's basically it's a state of equalizing pressures or gas pressures inside of the air. So if you have more moisture outside of the space than you do inside the space, the moisture will migrate on air particles and through the air in the opposite direction of pressure. To Drew's point, he just described how in a clean room, you want to maintain the cleanest space at the highest pressure and cascade air out in most cases. And when you do that, if you have a very humid environment on the outside, it will impact the humidity on the inside. So controlling the humidity, not just inside of the clean room, but outside the clean room can be important. Temperatures are a little easier to control, but you do need to make sure you understand how the moisture moves. And it's interesting as we've talked about, particles move back and forth too. And the dust and the tiny amount of these microns carry moisture and carry dust and carry viruses. They can actually carry things in the opposite direction of pressure as well. Not in large quantities, but you do have to understand that you do have no control over that. And it's important to understand that you need to then try and manage it through all of the design practices we're talking about.
So I know we spent a lot of time talking about mechanical solutions to clean rooms, but there are other design considerations outside of your mechanical solutions. So for instance, your architectural finishes will impact the clean room. Things such as FRP doors, or having FRP or PVC wall panels versus an epoxy painted wall, having a hard lid ceiling versus a gasketed and clipped ceiling tiles. Those are different design solutions that can be applied to clean rooms. I think the biggest thing here again goes back to understanding what's the protocol that the end users are using in that space when selecting these wall materials and the proper type of construction materials, because we need to make sure we're aligning with the cleaning products they're using, so that if they have a wipe down wall or what their procedures are, that the design solutions and the build out of the space can accommodate what they intend to do and how they intend to use that clean room. Another engineered solution could be door interlocks. So if you have an anteroom, putting in physical door interlocks so that the clean room door doesn't open until the previous door is physically closed. This will ensure that there's no instance where both doors are open and you've got a flush of air coming across from the dirty corridor into the clean room, or vice versa. And then lighting. So depending on what classification of clean room you're trying to meet, could impact how many air changes you need, or how many HEPA filters, diffusers you need, or diffusers in general. And what that could lead to is going to some sort of, if you need to increase the amount of HEPA filters in the room, you might have to put a teardrop light, which will sit on the ceiling grid, versus having a sealed gasketed 2x4 that sits within the grid. And those design considerations take into fact take into account total room size and air changes needed.
Drew, some of the other considerations on that slide, we talk about maintenance, and we talk about service and the idea of walkable ceilings, and are you going to be able to access things from inside or outside of the space? Those are all design considerations for maintenance and adjustment that need to be addressed when we're talking about the early design stages.
So now we talked about now that we've talked about different solutions that could be put into place, is when's the right time to use these solutions? Well, that's kind of all based on the guidelines for the clean room. So your design guidelines are the set of standards to minimize the contamination in the controlled environment. Different companies and universities have their own standards. These standards can either be performance-based or prescriptive-based. There's a big difference here. So performance-based is going to be kind of to meet a specific air change per hour. I'm sorry, prescriptive-based is to meet a specific air change per hour, whereas performance-based is going to be based on the allowable amount of a contaminant at a particular size. So various clean room classifications will allow for more or less contaminants at different sizes of micron of contaminant.
We had a question earlier, Drew, and I just want to touch on one thing in that last slide. The prescriptive piece of that, depending on the actual industry you're in, could be particles, it also could be active microbial growth, right? So bacteria or microbiological monitors can also be used to help you build in that performance-based solution in some cases. So there might be various performance measurements. So yeah, getting to the kind of the crux of it all, right? There's a bunch of standards that have been used over the beginning of clean rooms, very beginning, but it's really boiled down to the international standard, the ISO 14644 and the JIBS standard. Most all reference now the international standard and then add to it or amend it in some way, but it is the baseline for almost all of the standards for clean rooms. So the design guidelines impact construction and operation strategies, as I was just saying, they're not just performance, as Drew said, and they're not strictly prescriptive, but we want to make sure that when we design solutions that have both the construction requirements and operational strategies, and those two things need to play off of each other for every client, and it's different for every client. As Drew said, at the very beginning, we need to manage and understand the goals and operational goals of the process and what's going on in the clean room or clean space. Clean room construction. We're going to go through a couple different ways to put together a clean room, and then we'll go through a kind of a quick pricing analysis of kind of how that can impact a project or a client or a building. So different types of clean rooms. The first one we'll talk about is a temporary clean room, and it is exactly what it sounds like in the words. You could tilt up a ceiling with a couple posts and put HEPA filters and a light or two in there and get to a clean room classification that could meet the requirement of the science inside of the tent. It just recirculates back underneath of the curtain at the floor, equivalent to, say, a non-unidirectional low wall return. And it just goes back to the ambient environment in the space that it's in. That can be a dry space, or it could be a larger volume clean room. It could be a warehouse. So it really doesn't matter what environment is the background. What's important is what you're trying to achieve in that space, and whether you may even have an isolator in that space. So you could get to a pretty high level clean room class really fast, really inexpensively. And if it's temporary or even, semi long term, it can get through the stages until you get your permanent space built. So it has a lot of advantages. Stick built, actually the one you see most common in the US in most of our projects, mostly because people they have a fixed process or science they're going to perform in that space, and it gives them a faster path to construction, usually a little less expensive than the following two examples I'll give you. But you can lay out those walls. Sorry, Drew, one more second.
Very good.
No, it's okay. And the purpose there is you can have it all laid out, and as long as you don't need to move it or change it a lot, it is the most effective solution. We jump to the next one. So the modular solution is one that is applied pretty widely across the country as well. And there's a lot of manufacturers in the market now that are offering this system or systems like this, whether it's walkable ceilings, non-walkable ceilings, walls with chases, walls that are easily demounted and reconfigured. So there's a lot of options in this category of clean room. But the main thing here is you got to decide how many times do you really plan on changing it over the life cycle of that room. In which case, if that is needed and you think you're going to reconfigure it, it's a strong option and does offer a lot of advantages.
And the modular rooms, they're all prefabbed and then shipped to the site, ready to assemble. Whereas the stick built is raw materials on site that's built on site.
And it does give you a lot of advantages in terms of shorter downtime when you have to replace or manage the ceilings or change utilities in a wall or a utility panel on the wall or the ceiling. It's easier to manage those changes with a modular clean room than it is with a stick built. So then the last category, which is also really become more prevalent at the beginning of COVID. It's always been in the marketplace, but I think there's more of it being sold in the last 5 to 10 because of the demand for to actually have something located in a building really quickly. And you can put these into any warehouse. So they're manufactured, validated, certified, however you want to manage your science at the factory, put on a truck, shipped to the warehouse or larger volume space, rolled off the truck, put together, connect power, connect drain, whatever you might need, a couple of utility connections, and you're up and running. You can do some FATs and do final validation or certification testing when you get it in the space, but it's the quickest way to bring something up on site. And I've seen these connected with 5, 10, 15, or 20 of these things in very large warehouse spaces where you can build a containerized farm of a very nice clean room space. So yeah, the question everybody's probably thinking about but hasn't asked yet is, what does it all cost? And I like to say this comparison is based on systems without the utilities or the equipment, because that can vary widely across different industries. But if you had a plastic curtain clean room and you were tilting the thing up and you, depending on how many HEPAs and how nice the lights were, and I'll laugh what grade of plastic you use, you're talking in the $100 square foot range. And then you can get up into the scale of containerized systems and stick built where you could be, $2,000 and above, again, depending on the scale and size. I should say the $2,000 square foot range probably cuts into the modular range. And if you get into containerized, depending on the science you're doing, it can exceed that $2,000. So wide range, and happy to walk through that with anybody that has a specific application, because they vary so greatly. So testing and verification, we want to make sure that we understand what needs to be tested and verified. What we really get into is these situations where people want, I'll give you the example of, they might want a CGMP manufacturing space, but they don't need it for 5 years. So when you start talking about the testing and verification and the cost of that testing and verification and validation, it's very expensive. And a lot of times it's not applicable to what they're doing for the first few years of their space. And we tend to try and challenge our clients to make sure they understand what and when they should apply these things. But in the simplest form, to test and verify a clean room, you're measuring particles. And you need to be able to make sure that you either use a permanent monitoring system, depending on the reporting that you need to provide, if you need daily or weekly or hourly reporting for the science that you're performing. So you might have permanent monitors. And in the case of, a lower level verification of something that's certified annually or monthly, or certified on a restart of a project or specific science that might be turning over every month or every week, you can bring in temporary monitors and verify and make sure your space is appropriately classified and verified for the use that you have it. One thing that's interesting is this idea of particle counters and being able to do a handheld one. We recommend everybody that runs a clean room has a handheld particle counter in there. Depending on what the science is, depending on the volume of the airflow that you need to run through the particle counter, They come in various sizes and qualities and calibration requirements. But it's important to make sure that you do understand when and why you need that particle counter and have the appropriate equipment to do so. If you outsource it or have it internal, either way it works. Steady state versus operational airflow. This comes into play a lot when we talk about cGMP manufacturing and EU standards. They do require a steady state test without the equipment and people in the room. And it's just the background airflow. And it usually is the easiest test to successfully pass, but then you need to do the operational test that actually takes place when you're doing the process and have people and equipment running. That oftentimes in a very complicated space is nice to have the CFD, so you can have a proof of concept once you have the test done. We talked a little bit about validating clean rooms, but it's important to understand when you need to do it. Validating clean room starts with the design early, and then we talk about the initial testing. And then from that testing, you verify the particle counts over a specific period of time. You might, again, have bacterial counts that you need to do in the case of certain classes of EU and FDA standards. And after that verification, you can then compile all of your data and issue that. This is all part of a validation process that you would typically use an external third party, but some people have internal quality departments to manage this. And then you do all that certification and paperwork and submit it. And then you get registered and you can actually start using your clean room. Once registered, you get the validation and you get your certifications, wherever that may be in whatever industry that may be in, and you're ready for use. And so we'll jump into our next segment.
Yep. So the next portion of the presentation is about operating. So now that we've designed, built, validated, and registered, it's time to use the clean room. So the most important thing about using the clean room is the various protocols that go into using the clean room. So the protocols that are required criteria to continue to make the clean rooms operational. Your protocols are typically developed by someone from EH&S or from a quality team, and then they're handed to the various users of the clean room. This can be the scientists in the clean room. This can be the maintenance team maintaining the facility that serves the clean room, or it can even be the cleaning crew that comes in to the clean room. So the main purpose of these protocols is to protect the science and the people. But, you know, people should, Ray mentioned kind of the different sizes of microns earlier in this presentation. And one of the things that as humans, we shed skin cells, right? And that's going to be down on that 5-micron level of dust. So the protocols put into place for the people to get into these clean rooms and what they need to gown and how they need to wear their PPE is used so that we're not bringing in dirt or tracking any dirt into the space, and we're protecting the science that's going on in the clean room. Your protocols are your statements of procedures or your methods of procedures, SOPs and MOPs, depending on the industry that you're in. So couple of examples of protocols could be entering the clean room. So again, this is the PPE required. Do you need to change out of your street clothes prior to entering the airlock? Are you going in through an airlock, or are you going right into the clean room? What gloves do you need to wear? Do you need to wear booties? Are you in a Tyvek suit? Then in between sciences and experiments, how are you cleaning the clean room? What's the wipe down procedures that go into it? What are the cleaning materials that we're using to clean the space? When experiments are done, how do we do a full clean of the space? Are we gassing the space, or is it going to be all wiped down? And even there's protocols for maintaining the equipment. So this is inclusive of maintaining the lab equipment as well as maintaining the air handling units and the filters, and what's the protocols to change out the HEPA filters? Are the filters accessible within the space, or do we need to do a full shutdown of our air handling unit to maintain that equipment? So I know we talked about kind of at the beginning of the project, having that key meeting to involve stakeholders and understand what are the engineered solutions versus the protocol solutions that can take into place. And the main reason here is because engineered solutions can come at a cost. So we had a case where we had a project that there was a concern with mold and fungal spores drifting up to the surface of the science via the ceiling returns and exhausts. So the engineered solution in this instance would have been to put in low wall returns, but low wall returns are not cheap. They, require furring out the wall cavities so you can fit the duct, and it's more duct work. Sometimes it has to be stainless steel duct, depending on what's being cleaned, what materials are used to clean down the space. So what the end user ended up doing was they put in a protocol so that people entering the space had to change their shoes and their socks before entering the space to limit the amount of mold and fungal spores brought into the clean room. So go ahead, Ray.
I was going to say, we got a few case studies and some stories to share with you about things that went well and maybe some things that didn't go well. I did want to touch on one thing we talked about earlier. We talked a little bit about HEPA filters and how the air, before we jump into the case studies, I want to answer a question. And that was based on, the number of air changes and air velocities required in the EU standard for, class B, C, D spaces. And it varies greatly because what we're finding is everybody in the energy world is driving recirculated air or air changes to a minimum. And there are minimums, and you really need to make sure that depending on the science you're performing in those rooms, you have the appropriate air change to make sure you can maintain the particle count and the bacterial count. So what we find in the EU standard is, you get these, you get a minimum when you talk class B and C spaces, you could be as low as 60 air changes. But there are standards that reference 180 to 300 air changes. And those get very prescriptive to Drew's point earlier. We want to make sure that we're taking a more energy conscious approach, take more of a performance approach to that, and look towards being able to use something like an active monitor for both bacteria, kind of regular monitoring on bacteria and particle count monitoring so that we can have the ability to reduce the energy used. You can always go back to the prescriptive method, but I think the performance method is going to drive a lot of the newer clean rooms in the market as the newer environments get built.
Yep.
So
and another solution could be can that science be done in an isolator? And then you can have your background be under the isolator, and then the room background can be one grade up or one classification less to help minimize the bigger impact of energy at the air handling unit level.
Good point. So let's jump to the first. So this just happens to be an aerospace confidential client aerospace project we did where they were assembling satellites. And the tricky part in this early phase of the design was figuring out how they were going to drive a tractor trailer into a clean room. They really were driving a tractor trailer through the corridor that runs from east to west and dropping off the equipment. It was a non-classified space, but they tried to maintain it with HEPA filters at an ISO 8 level. And then inside of the assembly space that they had an ISO 7 environment. And what we did in that environment was originally we talked about things like raised floor, unidirectional top to bottom. And it came down to the assembly was going to be on platforms up above the floor, and with some testing and some verification, we were able to understand that they could use low wall returns and not have to go all the way to the raised floor solution and design a raised floor to a very high load capacity. So that was a success story in the world of aerospace and saved them a lot of money.
Here we have two case studies for cell and gene therapy clients. The first one on the top left, this is for Advanced Medical Partners. They're a CDMO partner focusing on viral vector manufacturing, process development, analytical development, and QC testing. And then the plan on the bottom right is for a confidential client, again, both pharma, both gene therapy clean rooms, but you'll notice one big difference between these two layouts, and this is, again, an architectural design consideration, is the use of the corridors. So on the top left, you have a supply corridor towards the bottom of the plan, and then you've got your airlocks into the clean room suite, and then you have an airlock into the return corridor. So earlier we talked about flow of air through clean room spaces, but there's also the flow of materials and personnel. So in this top left instance for AMP, the materials flow through the supply corridor, out through downstream, and through the production suites, and then out through the return corridor before they exit the clean room suite. Whereas in the bottom right, you have a common corridor to an airlock to each clean room suite. And in this instance, materials and personnel flow in and out of the space in the same pathway. So what you would do here is you'd make sure that all of your waste is sealed and all of your clean samples are sealed prior to entering that common dirty corridor.
Go back one sec, because I was going to answer another question, if I can interject here while you're on this slide. There was another question about velocities at the discharge of HEPA filters and how many HEPA filters you need for sealing coverage. And it really comes down to, again, as I said, how many air changes you want to drive in the space and how you want to be able to maintain or measure that particle count of bacteria or live cell count. So the standard really defines that you do it between 70 and 105 feet per minute. But what that really drives is the number of HEPA filters you're going to have. And I always say the smart solution is a lot like maintaining safety in front of a fume hood. Because if you're doing 75 to 100 feet a minute, you're maintaining a pretty constant flow at a reasonable velocity. So as long as you move with a methodical purpose and you don't jump around and run across the room or move real quickly, as long as you move slowly with purpose, the airflow will continue to move in the direction you want it to go, and the particles will generally carry with it. So that's really part of that standard of maintaining the face velocity off of those fan-powered HEPAs or filtered HEPAs at the surface of the wall or the ceiling. So the other kind of project that we're running into a lot lately is this quantum computing market, and we're doing a few of these types of projects. This happens to be one we did in our office in Denver, and the challenges here are not at the same level as manufacturing a drug or creating a clean environment for pharmaceutical, or even aerospace for that matter. But the idea that you need clean environments, you need clean background for things like lasers and NMRs and quantum computers, so that you don't have any noise in your respective data as you report out your new results. So it's becoming a new emerging clean room space in the world of quantum computing. Medical devices. This is something that falls sort of in line with pharma, but it's a different category because a lot of it's manufacturing, a lot of it's very large warehouse. And in the case of this plenum space, this walkable ceiling where they had all the accessible ductwork, we had to manage, in this case, similar to the aerospace situation, we had to manage multiple grades of clean room. And the interesting thing in this case, one room did require a raised floor, unidirectional flow from top to bottom, but the adjacent did not. So we ended up making sure that when the original science group said we want raised floor everywhere, we need unidirectional flow, as a good engineer, we challenged that question and said, so for this classification, you really don't need it, based on a standard definition of that classification of clean room. And then we challenged it and walked through where they needed the clean room. We made sure that the spaces they needed to be clean like again, back at the elevation of the space, you don't have to have the extra clean room classification if you don't need it, for example, at the floor. If it can be dirtier at the floor, you don't need the raised floor, completely unidirectional flow. So it gives you the opportunity, if you challenge your science and your assembly process, which can be up on a platform or up on a table, to be the clean environment, not the level at the floor where the particle counter really doesn't matter because it's not directly in contact with the actual medical device or that phase of the assembly of that device, which is the other part. You got to make sure you meet the standards you're building to. But we were able to save them a lot of money by challenging their own guidelines and pushing them in a direction that gave them a lot of capital to be able to use that towards other equipment that they needed. So I think it's kind of, that sums up where we really try and direct all of our clients, and in the process of starting from the goals, working through design, working through construction, possible techniques and design, so that we understand what our clients need, what are their business drivers at the very beginning of a project, to make sure that when we get from the very beginning, they have some vision of what the end looks like. And when you get there, there's this great satisfaction that we followed the path that we built for that client or for that project to get them to success. And hopefully then manufacturing or doing the science that they need in the room after their certification. So that's really the biggest takeaway, is to make sure you don't do things out of sequence. You challenge your own rationale, challenge your internal goals for timeline. When do you plan to need the space that you might need? And you're better off building something temporary or something less expensive, getting production up and running, and then building that future space when you have the revenue or when you have the appropriate actual requirement for that space.
So, right. Thanks. I just want to make sure we're at 10:51. Get some questions in here.
Sounds good.
There's one that's generally speaking for ISO 7 for electronics production. How does cost and schedule compare for stick build versus modular versus portable?
So the three scales come down to, I think I'm going to go back to something I said earlier, and it really comes down to how reconfigurable you need it to be. The stick built model will tend to be less expensive than modular or containerized. And it comes down to, if you're going to change your science or change your manufacturing, or you're going to add different lines of manufacturing or different lines of science, it could be different products in that world. In the ISO 7, you probably could go stick built. That would be my recommendation in that case. But modular systems are very, coming down in price. So depending again on the scale and size of the space, you could go modular. I'm not sure if that's a direct answer to the question, Dave, but with those little details I have, that's the best I can do.
Well, I'm sure everybody, and this is an invitation for anybody who wants to continue with questions for Ray and Drew. Please do. This is not a end all. That we hope to continue conversations with people as we do this. One other question, Ray and Drew, what is NMR?
Oh, NMR, nuclear resonance magnet. And that's because they use cryogenic liquids, and they use, they're measuring chemical makeup of a sample. They can actually figure out how many molecules or what chemical or what element are that build up what the actual sample is. That kind of gives you a sense of what scale they're measuring, to give you the fact that you do need a clean environment to use an NMR. They're not usually certified clean rooms in most NMR cases, but most NMRs are in a clean environment, a clean space. Probably to your point, Dave, this idea of clean spaces versus clean rooms. Clean rooms seem to always be defined as something that's certified. Something that's not certified also gets used. They use the term clean space more often.
So I have one question here. I'm interested in learning the assembly of all components that make up a clean room and maintenance best practices.
Okay, so the assembly of the components that make up a clean room is it could just be walls and the ceiling with HEPA, kind of like that typical. If you look at the simplest form, that temporary clean room, that's why I like that example in our presentation, because at its simplest form, that's all it is. It's a curtain that you can walk through, and then there's air that flows down that creates the clean space. But most of them are in the world of stick build or modular, and the cleaning practices come down to really what you're doing in the space. So if you have a dusty powdered substance type science happening inside the room, you're going to need to do a different cleaning procedure than if you were doing everything that was either liquids or out at the molecular level. You're not creating a lot of aerosolized dust or particles in the air. So you go from isopropyl wipe down to full CIP 1, 2, 3 mops inside of things, literally mopping the floor, wall, and ceiling every inch of it. So it varies greatly. But at its simplest form, it's a wipe down procedure to make sure you have a clean wall, clean floor, clean ceiling.
Okay. CFM requirements at each HEPA filter and ACH for the whole room for grade A and B clean rooms.
Yeah, I tried to touch on that a little earlier, and I think you get into this CFM coverage, or when you talk about roughly 70 feet or 75. We'd like to use 75 feet a minute at the HEPA filters, generally speaking. You get more ceiling coverage, which is better for the clean environment, and you get a lower velocity, which is a little easier to manage in terms of being able to move around in the space, as I said earlier. And we also like to use that also for noise. You know, when you get to, you know, north, anywhere around 100 feet a minute or north of that in some clean rooms, depending on the certification. In the case of EU, you really can't go beyond, you know, 106 feet per minute on those face velocities, but they're also noisier at that range, and you don't get as much ceiling coverage. So I tend to lean towards a few more HEPA filters in the ceiling at 75 feet a minute. And those air changes, you know, you're really going to see, again, you could be at 180 to 300 when you talk about B's and C's. We've done them as far down as 60. So it really comes down to what you're doing in the room. If you're doing something that doesn't need 180 air changes per hour, you can reduce that as long as you're maintaining the particle count and the bacteria count.
Great. And this is for everyone. The mics are open, so if anybody wants to just ask a question directly, please feel free to do so.
I did have one in a chat that mentioned, you know, just kind of this general difference between the FDA and the EU. The FDA standard does use the ISO classifications, and the EU uses the classifications, you know, the ABCD, as you mentioned in an earlier question, Dave. And the other big difference is that you're really looking at rest, you know, steady state, or you're looking at in operation when you do the EU standard testing for verification and validation. So there's a couple differences there. There are some deeper differences, and that's probably a topic for a whole nother hour, but at a high level from a clean room standpoint, those are the big differences there.
So is there another question? Is there a relation between ACH and ISO levels of clean rooms?
I think the relationship is that there are two different standards. We need to make sure that we're applying the right standard.
And each ISO range, each ISO level has a different range of air changes that they recommend meeting. So ISO 8 recommends meeting a certain range. ISO 7 is a higher range of air changes per hour, and so on and so forth.
And again, you go to aerospace versus pharmaceuticals versus food science versus microelectronics, and you know, they all come back to that main reference standard that I talked about, but they do apply things a little differently, and then they add additional requirements on top of it.
Great. So any more questions? Let's see. Thanks very much. I think anybody other questions? Because I think we
David,
met our goal.
David,
Sorry?
this is Frank Day from Cushman & Wakefield. How you doing, sir?
Sir, Frank.
Great presentation, guys. No disrespect to architects and engineers. I wish they did work more diligently on the operations side. It's difficult to get into things sometimes. Specifically, I have a question about filters. Is there a MERV rating on HEPA filtration? That this has come up a few times at some of our properties, and you know, trying to find HEPA filters for certain RTUs and air handlers is tricky. So certain MERV ratings have been recommended. I wanted to see if there was anything that you can shed light on MERV ratings and or HEPA filters.
Drew, we talked about HEPA filters being ultra pure and just pure HEPA filters, you know, 99.9 and then 99.999, I think. And if I'm misquoting that, I apologize. But to answer your question, or trying to be more direct, Frank, the MERV standard kind of ends at a specific particle count. It doesn't continue on through HEPA. HEPA becomes its own class of filter. So if they're asking you for HEPA filters, it's challenging to do at an air handling unit. We do it quite often. And it really comes down to horsepower and the appropriate pre-filters. But it's not something you can just throw into as a retrofit. To Drew's point, when you're in the earlier part of the presentation, we talked about retrofits versus new. And in a retrofit situation, you need to make sure that you have, you know, an additional half an inch to an inch to an inch and a quarter pressure drop to get into the detail, and that's going to equate to bigger horsepowers. And sometimes those fan motors and fans can take on that additional amperage or additional brake horsepower needs, and sometimes they can't. So there's definitely a fan study that would need to be done to verify if you can just add HEPAs. But the alternative to that is fan powered HEPAs in the ceiling to fan assist or basically push the additional pressure through the HEPA filter at the room or at the ceiling level. So that's an alternative if you run into that challenge.
And then you would pretty much swap out all of your diffusers with these fan filter units, 1 for 1.
Outstanding. Okay, thank you.
Hey, Drew, this is Stephen Peterson with 35 North. Thank you guys for a good presentation. The fan filter units that you were just mentioning that you could swap out for, would that be to provide the additional power to overcome the pressure drop across that local HEPA filter in a case where the air handler might not be able to push through?
Yeah, that's the exact reason. So if your existing air handling unit, has a lower external static, you need that additional fan at the fan filter unit to overcome the HEPA pressure drop, which will be at your diffuser level. So it'll all be within the room, and it's pretty much just a booster fan to get the external static across that filter.
Very good.
Yeah, Stephen, just to add to that comment, the air handling unit itself in a retrofit may not have the total CFM or air change requirement to meet the clean room class. So what we do in those retrofits is we add recirculated HEPA filters where the air doesn't even go back to the air handling unit. It just simply takes the air from the room, whether it's low or at the ceiling, depending on the clean room classification, and just recirculates it back in the room. So it's just simply spinning the air from the space through a duct at the low wall or through a duct at the ceiling back to that HEPA filter to maintain a higher clean room classification.
Gotcha. That does make sense. One thing I do appreciate the overview into other industries like aerospace and electronics. Speaking for myself, I've spent most of my time in the life sciences and pharma, and so you kind of get very focused on your particular requirements. But it's always interesting in reading the trade publications, you can see other applications. For unique solutions. And one thing I just found fascinating coming from a pharma environment where you're smaller rooms compared to aerospace. But recently I read an article about an aerospace application. I think it was for like a satellite assembly, and they had a very large area around a satellite work platform. And basically it was described as like a wall-less clean room where they had, it looked like a fan HEPA array above the work area, and the velocity and the air distribution was such that even without walls, there was, a very low probability of anything entering that airflow. It was like a waterfall, basically. And so it was just kind of interesting to see how a different industry could operate compared to the silos that some of us typically are used to.
No, I think, Stephen, that's something I was going to bring up earlier. Thank you for reminding me. But you mentioned one in a trade magazine where they actually did it vertically. There was one that I was a part of more than 20 years ago for NASA, where it was a horizontal unidirectional flow. And they had a big roll-up door in the side at the back of the room, and they would roll things in, close the door, let things get back to some semblance, some baseline clean room classification. And then they would slowly start taking the parts apart, and they would unpackage them. And as they unpackage them, they would move them from the back of the room to the front of the room. And it went from, an ISO 8 or almost unclassified space to the back, where they were taking cardboard and things inside of this space, which for people that don't clean rooms, you don't want a lot of cardboard or paper in there. And then you end up in the front of the room at an ISO, they were at a class 100 right at the HEPA wall. but they made sure they had lines on the floor. You could only do certain procedures behind a certain demarcation lines, and that's how they created the cleaner space as they moved through the actual satellite assembly, and they could remove the packaging parts and pieces at a time and then start to assemble. So it was a very unique process to the one you just described, and that was 20 plus years ago. So there's a lot of ways to do clean rooms. The room itself doesn't have to be all one classification and a lot of scientific applications.
It is interesting. And there again, from a pharma environment, we picture if you've selected the correct components and they were installed correctly and then cleaned, construction cleaned, and then placed into operation, and folks are just coming in clean gowns and proper gowning and so forth, you just assume that everything is okay. But if you look at it from the particles, like you talked about, where would particles travel if there were dirty areas, dust being stirred up, how would that travel? And one thing I realized in life science, there's a huge, there are procedural requirements to tell people to minimize talking, minimize motion, don't be horseplaying, when you got open containers. And that those are procedural concerns to minimize those particles swirling around to give the airflow a chance to do its job. So I just think it's fascinating when you think about the particles instead of the people.
I appreciate you emphasizing the procedural difference between the procedural solution and an engineered solution, right? You can move more air through that space, but you can spend a lot of money to do that. And as Drew and Dave and I pointed out, this idea that the more outside air you provide, especially if it's a toxic space, the more that costs. And by simply moving slowly and methodically and taking things in a very organized, slow motion, you can cut down on some of that. So you can save a lot of energy, a lot of construction costs, and a lot of operating cost by just simply implementing some smarter procedures. So you do need to do both, and you need to make sure that when you're dealing with each of these types of science, you communicate that at the beginning so that we come up with a solution that everybody is happy with at the end and can say, hey, this is exactly what we expected. Kind of back to that original, our starting thought is our end thought in these things. We always want to make sure we build with and design with, the functional solution in mind, that everybody gets what they need and it's a safe place to be.
And it's a combination of engineering controls and procedural controls.
Balance, the key. That's the key, balance.
All right. Thanks, guys.
Thank you.
Good questions.
Thank you,
Stephen. Thank you.
Anybody else? I think. Great. We got 12:08. Good. Have a nice lunch. Drew, thank you very much. Great job, guys. And thanks to you, everybody, for joining us, and looking forward to keeping in touch. And again, any questions, please don't hesitate to call Ray. And we'd love if you can take a scan and submit a online survey to see how we're really doing. This way we get better at what we do each time. Thank you, guys. Thanks, everybody.
Thank you.
Thank you. Thanks for joining us.